Good day, everyone. I am Norely Jan A. Borja, together with my members Miss Angel Bon and Miss Nina Katrina Gabitanan. So for tonight, we are assigned to present and critique the paper entitled Phylogenetic Analysis of HIV-1 Shows Frequent Cross-Country Transmission and Local Population Expansion. So this journal article was written by Mark Benepic, Anna Zukova, Manwong Eric Tang, Jacqueline Bennett, Paula Munderi, Kia Trungstrom, Magnus Gislen, Michael Warobey, Jens D. Lundgren, and Rasmus L. Marvik. This was written in the journal Virus Evolution, Volume 7, issued 2 December 2021, and published in the year 2021 of June 9. Currently, this journal article has a total of 6 citations with field citation ratio of 1.46, indicating a strong relative impact. However, its relative citation ratio consists of 0.35, meaning it cited 65% less than the average National Institute of Health funded paper. Now suggesting a lower influence compared to the high impact National Institute of Health benchmarks. In terms of Almetrics, it consists of two, indicates that a research article has received a small amount of online attention, typically from 1 to 2 sources. In general, the authors are currently affiliated with departments in Denmark under Research of Health Sciences, Infectious Diseases, Biomedicines, Bioinformatics, Ecology and other biological fields, and allied sciences. The main idea of this paper is about the HIV-1, which, according to the World Health Organization, remains a global health issue, having a claim and estimate of 42.3 million lives to date, and transmission is still ongoing across all countries. Specifically, this study on human immunodeficiency virus type 1 has geographically spread and genetically diversified into distinct viral subtypes. Due to high mutation rate, short generation time, and lack of proofreading and frequent recombination, this study will use a phylogenetic analysis to reconstruct the ancestry of virus and to better understand the origin of subtypes. Thus, the main objective of the study is to use the phylogenetic analysis of HIV-1 genomes coupled with geographical data to understand the global spread and evolution of HIV-1. Specifically, the study aims to, number one, characterize the genetic diversity of HIV-1 across a large, demographically diverse and geographically distributed cohort. The second is reconstruct the ancestral states of HIV-1 to trace subtype origins and patterns of cross-country transmission. And lastly, identify both frequent international transmission and localized population expansion of the virus. To achieve those objectives, the study followed a systematic multi-step approach starting with plasma sample collection from 3,785 ARC-naive HIV-1 plus participants in the START trial. Then, the researchers obtained ethics permits and informed consent from all the participants. Upon the collection, the RNA was extracted and amplified with two HIV-1 regions and sequenced using Illumina MySeq. Then, the reads will filtered against the human genome and aligned to the HXB2 reference to generate the consensus sequences. Then after that, the HIV-1 subtyping were identified using the REGA tool, and additional genomes from LAN-L database were incorporated. Then the genome sequence of the HIV-1 from the LAN-L with the subtypes of A, F, G, and CRFO1AE were downloaded and trimmed to match the start amplicons using MAFT version 7.453. Then after that, phylogenetic trees reconstruction using EXA ML and ancestral states were reconstructed using the PAS-ML with geographic annotations. Based on the results, the researchers construct a maximum likelihood phylogeny of all samples in combined dataset consisting of subtypes A, B, C, D, F, G, and CRFO1AE. From 3,785 HIV-1 plus STAR trial participants, only 3,197 samples with adequate coverage of GAG, POL, and ENV regions were sequenced and analyzed. Using the REGA subtyping tool, 2,354 were classified as the pure subtypes of A, D, F, G, and 843 as recombinants, including 147 CRFO1 AE. So as you can see in the phylogenetic tree, the branches are colored according to their subtypes. However, the outer color strips indicates if the samples are from either starts and LAN-L filtered web alignment. From the combined phylogenetic analysis of data from the start and LAN-A, maximum likelihood phylogeny of 5,133 sequences confirm a subtype clustering into monophyletic groups. So 2,501 starts HIV-1 sequence with a pure subtypes of A to G and CRFO1 AE with 2,632 representative sequences from the LAN-L filtered web alignment covering 72 countries. Notably, the analysis focused on subtypes A, B, C, and CRFO1 AE because other subtypes such as the D, F, G had too few samples to support reliable phylogenetic inference and ancestral state reconstruction. In the phylogenetic diagram, the circle represents genetic cluster of samples sharing the same geographic origin or the states. Then labeled shows the country and number of samples in each clusters, while the arrows indicate transmission events from cluster to another country. Arrow size and the number reflects the frequency of such transmission. Furthermore, multicolored circles labeled with 0 means multiple locations had similar probabilities of being the origin. So based on this phylogenetic diagram, a total of 3,155 subtypes B sequenced from 49 countries and found a likely US origins, having a probability of 89% supported by randomized reconstruction. While major cross-border transmission included 530 to Europe, Australia, then 179 to Latin America, and 61 return events. Notable cluster emerged in South Korea and China via Thailand, while Africa samples were all reintroductions. For the subtype C, a total of 949 subtype C sequenced and identified South African as the origin, with a probability of 99% confirmed by randomized annotation analysis. From the South African rooted cluster, there are four major transmissions seated from the large cluster in the India, Tanzania, Belgium, and Botswana. Then the largest Brazilian cluster included samples from both START and LAN-L datasets, indicating wide geographic spreads. Based on subtype a total of 337 subtype sequences were identified in Uganda as the origin. It consists of 99.8% probability that confirmed by control shuffling. From the Uganda-rooted cluster, 11 transmission were inferred, including one to Kenya that seated further spread. Then the Kenya link cluster is spread to Europe via two routes, through Tanzania, Cyprus, Greece, Portugal, and the second one is Congo, Ukraine, Russia, and Poland. Lastly, for the CRFO1 AE, a total of 439 sequences were identified in the Central African Republic as the origin, having a 64% probability supported by control shuffling. From Central African Republic, the largest cluster was originated in Thailand, having a 42 transmissions to small clusters outside Asia and large clusters within the Asia. Take note that most CRFO1 AE samples was originated from Thailand, having a 81% of those in the START datasets, highlighting Thailand as the primary source for these subtypes.
For the critic part, let's start with the title. So, the title of the study efficiently conveys the central focus of the paper, which includes the primary subject, HIV-1. The phrases cross-country and local population established geographical boundaries. This indicate a broader geographical scope as confirmed in the abstract, with about 35 countries involved in a clinical trial. In addition, phylogenetic analysis mentioned in the title is a method used to construct phylogenetic trees and reconstruct ancestral states. In the initial part of the abstract, it briefly introduces HIV-1 and its global spread that led to its evolution and diversification. In this section, objectives and methods are given emphasis. The objective, as stated, is the phylogenetic analysis used to reconstruct ancestral state in addition to the data from samples collected in both START and LANL HIV database. HIV-1 samples from 3,197 participants across 35 countries and 2,632 genomes are also indicated in this section. Although Table 1 samples under START are lesser than the participants mentioned in the abstract, only 2,501 samples are reflected in Table 1 because of its low quality sequences and ambiguous subtype classification. Results are briefly stated in this section, also including the most homogeneous subtype population in areas where samples showed greater diversity. The subtypes with the largest cluster are emphasized as the focus of HIV-1 transmission pattern investigation. These subtypes include A to C and CR01AE, which are all given separate discussion and diagram of transmission in the result and discussion, while D to G and other recombinant HIV-1 subtypes were not given much emphasis in the abstract. To sum up, abstract of the paper serve as concise summary of the entire study that help reader understand its relevance to their interests. For the purpose and objective, the article maintained a consistent alignment with the study's purpose and objective. As discussed in the introduction, the study addresses concern about retrospective nature of phylogenetics in the investigation of HIV-1 transmission across country and within local population. This approach highly depends on the comprehensive sample collection, which require the involvement of multiple countries to gather all genotypic states, including ancestral genotypes. To address such challenges, they incorporate specific methodologies which include the involvement of large datasets, screening, sequencing, reconstruction, and cross-validation. These methods are all grounded to the study's primary objective to determine the transmission pattern and pathways of HIV-1. In the discussion and ideas, the paper provides comprehensive discussion supported with relevant ideas and related literature to reinforce the study. However, although subtypes A to G and CR01AE were all detected from both START and LANL datasets, only subtypes A to C and CR01AE are given emphasis due to their large cluster and relevance in transmission and geographic analysis. In contrast, subtypes D to G were not emphasized due to their low frequency in both datasets, limiting their contribution to reliable inference. Another concern arises from an equal collection of samples across countries. Imbalanced sample size can obscure the actual regional trends, which may influence the frequency of HIV-1 subtypes. Low frequency of subtypes D to G may be attributed to their higher prevalence in undersampled countries which were insufficiently represented in datasets. Therefore, ensuring sufficient sample size per region should be practiced to avoid biases in potential underrepresentation of the region. On the other hand, while study investigates some recombinant subtypes of HIV-1, recombinant subtypes were not extensively analyzed due to their complexity. However, despite the complexity, inclusion of more than one recombinant subtypes is advisable to enhance datasets, which may be essential in accurate and real-time reconstruction of their phylogenetic tree and analysis. Overall, the study presents clear, concise, relevant data. The title also clearly states the study's objective, while abstract effectively summarizes the entire paper, including the result of the investigation. Also, references, ideas, and methods are all relevant and directly aligned with the study's objective.
Good evening, everyone. I am Ninya Katrina Gabitanan, and I'll be continuing our critic discussion by addressing the following questions. Number 1. Are the author's statements clear? Some statements in the study could be more precise by defining the key terms. For example, the continuous transmission. A clear phrase would specify whether transmission occurs in sustained chains or isolated introductions. Next, providing the quantitative context. For example, specifying subtype prevalence percentages. It would be clear if the study explicitly stated which subtype is dominant compared to the broader diversity in African samples. And lastly, by clarifying methodology. For example, explaining how transmission was delineated. A more precise description of the methodology used to analyze transmission would enhance clarity. Number 2, what underlying assumptions does the author have? Here are some underlying assumptions the author makes in the study. First, phylogenetic analysis accurately reflects transmission patterns. The study assumes that maximum likelihood phylogenetic analysis is a reliable method for tracking how HIV-1 moves between populations. This suggests that genetic relationships can be used to infer transmission events, even though they are not direct observations. Second, geographic data enhances transmission understanding. The study integrates location-based tracking, assuming that geographical data aligns with genetic sequences to help identify cross-border transmission. This means the authors believe the origin and spread of HIV-1 subtypes can be inferred through mapping techniques. And lastly, large sample size improves accuracy by analyzing 3,197 genomes from 35 countries. The authors assume that a diverse dataset strengthens conclusion about global HIV-1 transmission. However, this study relies heavily on clinical trial participants, which may not fully represent broader HIV-infected population. Number 3. Has the author been objective in his or her discussion of the topic? The author generally presents the topic objectively by using data-driven analysis and scientific methods to examine HIV-1 transmission patterns. However, certain assumptions, such as the emphasis on cross-border transmission, might reflect a scientific perspective that could be further tested. Additionally, the selection of data and regions studied could introduce some bias, as certain population might be underrepresented. While this study is largely data-driven and objective, addressing these areas could help present a more balanced perspective. Next, are the experimental methods described adequately? And are the study designs and methods appropriate for the purpose of the study? Well, this study provides a detailed description of its methodology using maximum likelihood phylogenetic analysis combined with geographical data to track HIV-1 transmission. With 3,197 genomes from 35 countries, the dataset is broad and diverse. However, because this is a descriptive study, it does not follow a traditional experimental design, but instead relies on genetic sequencing and computational modeling to infer transmission trends. Regarding the study design and methods, they are generally appropriate for analyzing HIV-1 transmission patterns. The phylogenetic analysis effectively maps how the virus spreads across borders, and the use of genetic sequencing provides strong evidence for transmission dynamics. However, there are some limitations that could be improved. First is the computational settings are unclear. The study lacks details on software parameters, making replication difficult. And lastly, the sample selection bias. Most participants came from clinical trials, which limits population diversity. The statistical methods used in the study are generally appropriate for analyzing HIV-1 transmission patterns. However, while the methods are well documented, some details such as specific computational parameters or data filtering criteria could be expanded to ensure full reproducibility. If a researcher wanted to duplicate the study, they might need additional information on software settings and sample selection criteria. In addition, some parts of the study repeat the same ideas, especially when talking about cross-border transmission and HIV-1 subtype diversity. The introduction explains that HIV-1 spreads across countries and has different subtypes. The results section repeats this by showing which subtypes are common in certain regions. Then, the discussion brings up the same points again, instead of focusing on what the findings mean. To make this study easier to read, this section could be more focused by introducing the topic, showing the results and then explaining why they matter without repeating the same information. In conclusion, this study explored HIV-1 transmission patterns by using phylogenetic analysis to examine how the virus spreads both across borders and within local communities. It highlights the importance of subtype diversity and cross-country movement in shaping the epidemic. From a critical point of view, while this study presents useful findings and applies strong methodology, it is weakened by repetitive content and a lack of deeper interpretation in the discussion. The study also misses the opportunity to connect its results to a real-world health strategies or offer clear recommendations. To improve the clarity and impact of the study, I recommend streamlining repetitive sections, particularly regarding cross-border transmission and subtype diversity. The study would benefit from a more focused discussion that explored the practical implication of the findings for public health, offering concrete recommendations for interventions, surveillance, or policy would strengthen the study's relevance and applications. Despite these limitations, the research contributes valuable data to our understanding of HIV-1. Its findings can help guide future studies and improve public health planning. Understanding how HIV-1 spreads at both local and international level is essential for designing effective prevention and control measures. And that concludes our critic discussion on the study. Thank you for your time and engagement.